Thank you so much, Amy. Our next speaker, Brian Fiske, is from the Michael J. Fox Foundation for Parkinsonian Research, and he'll talk to us about advocacy on behalf of those with Parkinson's disease.
All right, thanks so much, and thanks again for letting me participate in the meeting the last 2 days. It's been very informative for me. As I get started here, I guess I would just want to clarify, I won't really be talking about the patient perspective specifically around precision medicine or precision modeling. I think actually a lot of the points that Daryl raised, I think are probably true for people with Parkinson's as well. They're sort of understanding even what precision medicine is or precision modeling. What I really hope to convey, though, is how sort of the concepts of precision medicine and modeling are really sort of influencing how we understand Parkinson's and ultimately how we are pushing to develop new treatments for patients. And I think that's really where a lot of the impact is happening. Let's see, how do I advance? There we go. Okay, so I guess I'll start first with just for those of you who might not know the organization I work for, we are the Michael J. Fox Foundation for Parkinson's Research, and we are the world's largest nonprofit funder of Parkinson's research. We have a very clear mission really given to us from our founder, which is really about finding a cure for Parkinson's through really an aggressively developed and funded research agenda, and ultimately working to ensure development of drugs for those with Parkinson's today. So we've been around since late 2000, actually again funded by actor Michael J. Fox. We're headquartered in New York City. We're a public charity supported completely by donations from a variety of different individuals, groups, and other nonprofits. We've been successful since our founding in 2000. Have been able to deploy close to $750 million in research programs since that time. We fund a variety of different groups, academics, actually for-profit companies as well who are doing Parkinson's disease drug development. We also fund internationally. A good third of our funding goes to projects that are led by investigators who live outside the U.S. So we really go wherever we can to find the best research. We use a variety of different strategies and sort of thinking about how we can promote research and drug development in Parkinson's. Many in the field know us for our funding programs, and we have a number of funding opportunities throughout the year for people doing research in Parkinson's, doing drug development in Parkinson's disease. But probably in sort of germane to the conversation of the last 2 days, we also spend a lot of time developing tools for people in the Parkinson's research space, and these a variety of different tools from sort of protein reagents, sort of your typical laboratory reagents, antibodies, things like that, but also research models. So we actually spend a lot of effort building new mouse models, rodent models of the disease, and working to make those as freely available as we can to the community, whether you work in a nonprofit or a for-profit institution. And really trying to break down those barriers. We also spend a lot of our time and effort following people with the disease, which is obviously probably the best model of Parkinson's disease out there. And we've funded and supported a number of different cohort efforts, prospective cohort efforts. I'll talk a little bit more about some of that in a moment. But really with the idea of collecting deep clinical data and biological data on people with the disease and really using that to inform our understanding. And all these data are available. You can download them from the website and also request access to a number of the different biosamples that we've made available. More recently, we've also gotten more involved in sort of the advocacy policy side of Parkinson's. We've always been very dedicated, obviously, to the research endeavor, but over the last couple of years, we merged with a group called the Parkinson's Action Network and combined forces really with the idea of better advocating for and targeting specific policy issues that impact our ability to do research in Parkinson's disease. And you may see our name popping up more and more in sort of areas like this in the next few years. And again, as I said, we work with a whole variety of different stakeholders, obviously the patient community, the research community, drug makers, regulatory officials, and pretty much anybody involved in the Parkinson's space. So then really for the purposes of the talk today, it's probably good that I'm making this statement, but I think in Parkinson's disease, we obviously feel that we're ready, I think, for a precision medicine approach, and I'll talk a little bit more about that in a moment. But really, I think that there are a couple of major key drivers that are making us push for this today. For one, we know that people with Parkinson's disease vary greatly in their disease, the progression of the disease, the severity of their symptoms from person to person, the response to treatments, suggesting that there clearly are sort of fundamental subtypes within the disease that might be impacting our understanding and ultimately how we can treat the disease. We know already that the cause is likely multifactorial. We know there are certain genetic forms of the disease that have been identified. Unfortunately, a large percentage of the cases out there are still idiopathic. We don't really know the cause, but as we uncover the genetic forms, it's pointing us to cellular pathways that we think might be more relevant and gives us then an opportunity to sort of tease apart the biology of these other idiopathic cases. Through biology, obviously we've been able to sort of tease apart some of the cellular dysfunction we think might be linked to Parkinson's disease, pointing to a whole variety of cellular systems, many of these not necessarily unique to Parkinson's, but impact on mitochondrial function, cellular protein handling pathways, lysosome biology, a whole variety of different systems involved in cell health that we think could be underlying certain forms of Parkinson's, and sort of teasing out whether these are divergent, sort of parallel pathways leading to ultimately to Parkinson's, or whether these are sort of converging on common pathways is still certainly an open question, but clearly there are multiple sort of mechanisms involved in Parkinson's. And also, too, I think even just thinking about what is Parkinson's versus some of the other brain neurodegenerative diseases, and thinking about the shared biology that we see across some of these diseases, and potentially even considering whether we should be taking a page from the cancer field and sort of rethinking how we classify and categorize a lot of these brain disorders into more around their biology and less around their specific clinical sort of descriptions. And it's, again, I think just as I said, it really is a time for us really to rethink again this sort of clinical focus that we've had. So I put this in there, I put this slide in because I kind of liked one of the concepts that was raised yesterday around the sort of concept of the map, sort of the Google map approach to thinking about diseases. And, you know, I think we can apply this obviously to Parkinson's as well. But we sort of think about the different layers of information and data that we apply to our understanding of disease, from the sort of molecular understanding of the disease all the way up to ultimately sort of the map, if you will, of the patient's daily experience with the disease. And I think the importance of sort of being able to link all of those different sort of layers of the map into a sort of a clear understanding of disease and ultimately disease subtype. It is interesting that actually, and then sort of coincidental, that this is actually the 200th year sort of anniversary of sort of the first formal description of Parkinson's disease made by neurologist James Parkinson's back in 1817. Back then he didn't call it Parkinson's, I guess he wasn't that ambitious, but he called it a shaking palsy, but ultimately others ended up giving the disease his name. Since that time, though, we really haven't changed in how we think and sort of classify Parkinson's. It's still largely diagnosed by, particularly its motor symptoms, which are the ones that I think most everybody sort of appreciates and understands. Sort of the slow movement, bradykinesia, the rigidity, the resting tremor that sort of most Parkinson's patients ultimately exhibit, and some postural instability that some patients exhibit as well. And really, this is sort of the defined features, the sort of the clinical diagnostic features of Parkinson's disease still used today. We do know that Parkinson's has a whole variety of other quote-unquote non-motor symptoms, so it's not just the motor symptoms. Cognitive impairment, actually a large percentage of Parkinson's patients ultimately go on to develop some form of cognitive impairment later in this stage of the disease. Mood disorders, autonomic dysfunction, loss of smell is actually a very early sign, even sort of prodromal to the motor symptoms. REM sleep behavior disorder, another sleep issue that's found often pre-motor to the disease. And again, just sort of a clear understanding that Parkinson's isn't just about the motor features, that there's sort of the spectrum beyond that as well. But again, 200 years later, we're still really using this clinical understanding and definition of the disease, and I think that's what we're sort of struggling with today, and ultimately I think where we're finding precision medicine may help us move to in the future. We do obviously know more than just the clinical symptoms. We've learned a little bit about the underlying pathology, and I would say, you know, mostly what is driving that understanding is around two key features, and this is also what's driving mostly how we treat Parkinson's today and how we're sort of currently developing treatments for it. And this is really around the concept of loss of dopamine in the brain, loss of dopamine-producing cells in the brain of people with Parkinson's, and an accumulation of an abnormal accumulation of a protein called alpha-synuclein. We do know that, again, that Parkinson's, at least the motor features, are caused by this loss of dopaminergic neurons in the substantia nigra. And you can see, here for a Parkinson's patient, sort of that clear, obvious loss of these melanin-containing cells in the substantia nigra. We know that there is a pathological accumulation of, in cells called Lewy bodies in the brain that can also be found in those same regions, and that ultimately, and this was discovered really about 20 years ago, that the main protein component of those inclusions is the protein alpha-synuclein, and that's really, again, I think, galvanized and driven at least our current understanding of the disease. These are obviously important criteria for confirming actually whether you have Parkinson's, and actually these are used for neuropathological confirmation of the disease. But there actually are a variety of other pathologies and brain cell degeneration that it can be exhibited in certain forms of Parkinson's. And again, so as we sort of expand the picture of Parkinson's, we're appreciating that it is not just a single sort of simple description. But again, these are really what's driving our sort of ability to treat the disease today. Another aspect that when we think about sort of precision modeling, sort of understanding of Parkinson's, is the fact that it's not sort of a single disease that sort of is all or none. It really progresses over time, and you can kind of see that here with people who, again, currently defined by motor symptoms early on. Predominantly motor symptoms are the things that are driving their disability. There are treatments that can help with that motor disability for the most part in those early stages. But the disease progresses ultimately where you have late symptomatic sort of changes in the disease where those motor features become less the problem and it becomes more the spectrum of non-motor symptoms. And how we sort of think about treating those individuals at those stages is obviously an important question, but I raise it today only because it also is important to think about how we model these different features of Parkinson's disease in the lab to be able to make better treatments. Also thinking about pre-motor sort of Parkinson's disease and sort of the features of the disease that happened before that current sort of clinical diagnostic line is crossed, and how we think about those early stages of Parkinson's as well is really important. I put this up there because this is sort of an interesting, this is data actually from a patient cohort study called the Parkinson's Progression Marker Initiative. It's a large prospective study that we funded for the last several years. And these are sort of shows the example of how relying on sort of clinical subtyping can sort of lead you down a potentially problematic path when we think about it in the case of Parkinson's. So for a number of years, neurologists have tended to classify their patients along a couple of different clinical subtypes. There's a subtype that oops, let me go back. How do I go back? There we go. There's a subtype that they call tremor dominant, which is particularly found in Parkinson's disease and sort of more your traditional sort of tremor-related Parkinson's. But there's also a form that is called postural instability gait disorder, which is less tremor, a little bit more gait problems. These people tend to be a little more akinetic in their disease course, and then there are people who sort of fall somewhere in the middle. So in the study at baseline, these were people who were newly diagnosed with Parkinson's disease. The researchers, what they did was they classified them based on their baseline clinical subtype. We then looked at them a year later and reclassified them to see how stable those definitions were over time. And what we found after even just a year of early Parkinson's, again, these are people newly diagnosed who've been followed for a year, is for the most part, people who were tremor dominant at baseline tended to stay that same way. There were some people who sort of turned into postural gait instability folks or sort of went somewhere in the middle. But what was really interesting, though, to see in the posture gait people, many of those people actually started developing more tremor-dominant Parkinson's disease. And sort of people in the intermediate region, maybe not surprisingly, sort of went one way or the other. But what this, I think, really reflected was that this sort of concept that you could sort of just by clinical symptoms alone tease apart different forms of Parkinson's probably was not going to necessarily always help you, especially when you start adding in time and progression of the disease to the equation. So how have we been modeling this in the lab? And I think it's safe to say we have plenty of models, attempted models of Parkinson's disease that have been made, and they've been categorized mostly around two general types. There's sort of the environmental trigger type models and sort of the genetic-based models. Those in the environment include models that use various neurotoxins really to knock out the dopamine system. MPTP is a classic example, 6-hydroxydopa, some actual pesticides pesticides as well. Rotenone and paraquat have been used as a way to get rid of the dopamine system. And again, these are good models of sort of the symptoms of Parkinson's. You get sort of motor features that are somewhat, resembling those in people with Parkinson's. Other triggers people use, inflammatory signals and things like that, ways to sort of impact the dopamine system in particular. But I think the real interest is, of course, these genetic models that have been developed through a variety of different strategies, knockouts, knock-ins, transgenics, also use of viral vectors to deliver some of these specifically to certain regions of the brain. And we do, as I said, know that there are at least a handful of genetic forms of Parkinson's, synuclein, LRRK2, GBA1, PARKIN, PINK1, DJ1, VPS35 are a handful of the sort of the more higher risk genes. There's a whole list I could get into potential sort of risk, more modest risk genes as well that have been pulled out of GWAS studies. But it's safe to say that these are sort of the prime models people are really focusing on and thinking about how to model Parkinson's in the lab. I put this in here really just as a reminder to talk again about how the importance of alpha-synuclein in really driving a lot of our understanding, and this is really a big area for us, and especially in how we sort of can measure alpha-synuclein in people with Parkinson's disease. We are trying to measure it everywhere we can. We actually have a major study right now that is seeking to do that, and just again really trying to demonstrate the importance of how correlating sort of synuclein in people with Parkinson's is such a driving factor for being able to model it in the lab. How that is ultimately, though, being used in the lab, of course, though, is generating models of alpha-synuclein pathology, and there are some genetic models that use, again, some of the genetic forms, mutations in alpha-synuclein, but this model in particular has raised a lot of interest. This is based on an idea that synuclein pathology might actually physically spread from cell to cell in the brains of people with Parkinson's, and there's a lot of sort of background and history on this, and as well as some controversy I won't get into, but basically this model is being used now quite commonly as a way to model this spread of alpha-synuclein pathology, and using that then as a way to sort of ultimately test potential drugs that might have impact. A couple of other models, though, and then I have one more slide after that, are really, again, around the genetics, and I think this is, again, showing the excitement that we're starting to build as our genetic understanding of disease is ultimately helping us understand the biology of Parkinson's. A model, a sort of understanding of GBA linked to Parkinson's disease is one clear area. And we've been generating some models to better understand this, the activity of the glucocerebrosidase gene in models of Parkinson's. But also, too, another gene called LRRK2, which I think is really interesting. It's still relatively rare in the sense of genetic forms of Parkinson's, but has a lot of interest right now. And through some work that we funded in the last couple of years, we were able to identify a family of proteins called RAB-GTPases that are physiological substrates of the LRRK2 protein. It's a kinase. And we've been able to use that now to sort of model and measure the sort of phosphorylation of RAB proteins as a potential readout in model systems of LRRK2 Parkinson's disease. And so there's a lot of excitement building here on our ability to actually sort of take what ultimately is a genetic form of Parkinson's, but now has been linked to a cellular readout. And the idea is that that cellular readout might be something we could even look at people with idiopathic disease to see if their sort of RAB protein systems are showing similar changes. And I think that's really starting to excite the field right now. I should add, before I leave this slide, one of the probably closest examples we have to use of precision medicine in Parkinson's right now is the fact that there is actually a trial of a GBA1 pathway targeting drug by Genzyme right now that's specifically targeting people who carry mutations in GBA1. So that's kind of one of our first attempts to sort of use, in this case, genetic screening as a specific criteria for testing a drug in Parkinson's. So I'll end here, my last slide, really again just with a call out to say that as much as we can try to model Parkinson's in the lab, for us, one of the most important things to continue to be doing is modeling Parkinson's disease in the best model of Parkinson's disease, which is people with the disease. And so we've spent a lot of time and effort. We have a major initiative called the Parkinson's Progression Marker Initiative. Happy to talk to anybody more about that if they want. As well as sort of more emerging technologies and sort of use of online cohorts. We also have a sort of emerging effort called Fox Insight, which is, again, sort of an online version of PPMI, using a whole variety of different online surveys and collection tools, including use of wearables and things like that, to collect as much data from people with the disease over time as we can, and ultimately using that, again, to come up with a better informed understanding of what is Parkinson's so that we can better treat it. And I will stop there.
Great.